Lo importante es concentrarlo aquí o en esa So do you see it better from that location? Right? Okay. I'm happy. I'm happy. So the objectives of this seminar is I want you to understand this new paradigm of molecular target therapy because it's very important in medicine and it's evolving very fast. I would like you to have an in-depth knowledge about what are monoclonal antibodies, what is their mechanism of action, a little bit of pharmacogenetics related to monoclonal antibodies, and applications and new developments associated with this monoclonal antibodies, like new strategies, right? And these two aims will be covered in the next seminar, next week. Okay, so I'm not going to talk about that. Okay, so the use of immunotherapy is nothing new. In fact, Ehrlich used, by the end of the 19th century, used sera against diphtheria and tetanus. And it was effective. It was immunotherapy. And for that, he won the Nobel Prize. But this kind of antibody were polyclonal. And that's very important to distinguish monoclonal from polyclonal. Polyclonal means that what you have, it's a mixture of different antibodies recognizing different epitopes of an antigen. Okay? Situation is different to the monoclonal antibodies, where you specifically target just one epitope. Just one epitope. This monoclonal antibody, we can make the abbreviation by saying MAPS, can activate, like being an agonist, or suppress, acting as an antagonist of a molecular function, because it interferes with the specific molecules, and that's the difference. So here you can see, for an antigen, a monoclonal antibody will only recognize one type of epitope with a specific molecular structure. The synthesis is not new either. It was in 1984 when the one that discovered this way of synthesizing monoclonal antibodies won the Nobel Prize. So this is quite important. And how do they do it? Well, basically, I mean, if I want to target a specific antigen, let's say an altered molecule that is responsible for a disease. Okay? So what I should do is to take this protein, human proteins, because we want to treat a human being, a patient. We use this protein and we inject it into a mouse, a rat, a goat, a rabbit, an animal. This animal will react against this protein by synthesizing monoclonal antibodies. First, plasma cells expressing monoclonal antibodies. Polyclonal. These are polyclonal antibodies. And these cells will die, so I just can get few antibodies. But if I fuse these plasma cells with a tumor cell from cancerous plasma cells, I can create what is called a hybridome. And hybridome can live forever, as long as I take care of this cell line and I put ingredients to keep growing and keep living. So this is a immortal cell where I can collect in the supernatant all the antibodies that it generated. If I dilute this collection of plasma cells, having just one cell per plate, all the antibodies that are synthesized will be monoclonal antibodies. Okay, so these are the guys who discovered this kind of procedure for synthesizing, for creating monoclonal antibodies. So that was quite a long time. If we think about 1984 is quite a long time. So why we haven't used this monoclonal antibody in therapeutics during that period? Because monoclonal antibodies in therapy it's very new. It's something really new. It could be like, I don't know, maybe 15 years from something from the 21st century. So why it was so complicated at the beginning? Well, the main problem is the immunogenicity, adverse effects. Because you think that these antibodies have been produced in a mouse, and this mouse will synthesize mouse monoclonal antibodies, and we will react against that because it's a foreign protein for us. We don't have antibodies from mouse, so our immune system will react against the therapy, and this therapy won't be effective. There will be very low half-life in plasma. It will be cleared from our blood if someone injects us antibodies from a rat, even though we'll target the molecule that is altered and responsible from our disease. Okay, do you understand that? Okay, so, and that's another point. The point is that, let's see if I can use some. The other thing is that this molecule made in mice can only be recognized by mouse immune cells. So there are low effector functions through the Fc receptor. The Fc portion of the antibody will be recognized by a cell expressing a receptor, Fc receptor. You know that, okay? So, but this connection will be effective in a mouse because it's designed for being recognized by mouse cells expressing Fc receptor. But not if this molecule, this antibody has to connect with the human immune system. So there's a loss of efficacy of this monoclonal antibody, right? So what during all this year, from the discovery to the application in therapy, what researchers have been doing and pharmaceutical companies is to remove the murine part, the part from the mouse, in that antibody using recombinant DNA and substitute it by human. So here we have fully human monoclonal antibodies that will recognize human altered molecule, and this can be useful for treatment, and it has an Fc portion compatible with human cell expressing receptor against the Fc portion of the antibodies. So that took some time. Okay. So this is the process. It's in Spanish. You can understand it better. You have to again inject the antigen, but the thing is that you can remove by recombinant DNA you can substitute the murine part, the part from the mouse, with a human part, and then you'll make a recombinant human monogene that, when expressed, will give you monoclonal antibody, a human monoclonal antibody that can be grown forever, in theory, forever, and that's because it's so profitable for pharmaceutical companies. It's a very good business. to synthesize monoclonal antibodies. So, as you can imagine, each monoclonal antibody has its own target. And you can see it here. These are different monoclonal antibodies with a target. And this target is playing some kind of role in different kinds of diseases. We are not talking just about cancer, but also about autoimmune disease, like rheumatoid arthritis. And there's a whole spectrum of different diseases that can be treated with monoclonal antibodies. And this is based on this concept of molecular target therapy. Here you can see some of them. These are basically to treat cancer, but here you see Crohn's disease, ulcerative colitis, even hypercholesterolemia can be treated with monoclonal antibodies, and a whole list. So, what we have now, we have developed very high throughput techniques that can allow us to know all the molecules that are altering a given disease. And this is an example of what has been done in cancer. So you can see here different types of cancer and different types of genes that are mutated somehow in its specific cancer. So this information is really valuable, it's priceless. We can use this information for treating patients. Why not? Why not? So the idea is, why, if we know that the target is different in this tumor than in this other tumor, why are we going to treat with the same way, with the same weapon, with the same chemotherapy? So that's because target molecular therapy is so emerging in medicine treatment. It's really a revolution. And so monoclonal antibody will be part of this concept, but you also have to think about another molecular target therapy like tyrosine kinase inhibitors, proteasome inhibitors, and even gene therapy and gene silencing, but this is more still in preclinical study. One funny thing about the monoclonal antibody is the nomenclature. You have to think that every active principle ending in math, it's a monoclonal antibody. That's easy. And also depending on how much content of the antibody is from mouse or human, you can have like different parts in the word. So if it has an O, it's because it was synthesized in mouse. If A, it's rat. U, human. If it's chimeric, means that it has half mouse, half human, and so. And there's also some kind of prefix indicating some characteristic of this disease or the monoclonal antibody itself. You also have to think that not all biological therapies are monoclonal antibodies. These are monoclonal antibodies. These are targeting tumor necrosis factor, but also we have some other which are not monoclonal antibodies, but are also recognized in TNF, tumor necrosis factor. And but you can see by the name that it's not a monoclonal antibody. Here, etanercept, it's also targeting anti-TNF, but instead of using the variable region of a monoclonal antibody, we'll use a receptor, the TNF receptor. By recombinant DNA, we can cut pieces of DNA and put together one piece coding the Fc portion of the immunoglobulin and another part of the gene coding the receptor of TNA. So this is another way of binding targets, of binding ligands. Any question? Do you understand? More or less? So the mechanism of action. So now we have seen how monoclonal antibodies can be produced, and now we are going to see why they are so effective. They are so effective because they can interfere with molecular pathways. You have seen in our class about neoplasia that you have a receptor on the membrane that receives a growth factor, it dimerizes and then cross-phosphorylates. So we can interfere with the dimerization here. with a monoclonal antibody that can recognize one monomer of the receptor. So this receptor cannot find the other part to dimerize, so the pathway will be blocked at this point. We can also find monoclonal antibodies that can bind the growth factor or the soluble factor, the hormone. That's that way avoiding the pathway towards the nucleus. These monoclonal antibodies, and this is typically from the immune system, every time you have an antibody that can be recognized by a cell, these cells can induce the apoptosis of the cell expressing the target, the antigen. So this is another way. And there is also, by certain types of cells like monocytes or even natural killer cells, they are able to recognize by this Fc receptor an antibody, and then exert what is called the antibody-dependent cellular cytotoxicity. So these target cells will be killed by the products that are contained within the natural killer cell, or the granulocytes can make phagocyte the target cell. Okay, and also knives. So these are things that we have seen in class. So it's I think it's a nice content for you. All this topic, because you see things from the part of the immune system pathology and from neoplasia. Okay? And these antibodies can also bind the complement, so it can cause the complement activation and the formation of these pores on the membrane that can destroy the cell from inside. Okay, so this is an explanation. You will have it about what I've explained with the figure, with the previous figure, and but still there are some problems of immunogenicity, even if they are human. These monoclonal antibodies can induce what is called anti-drug antibodies, ADAs, and this is a mechanism of secondary resistance. So initially the patient responds, and then you find out that there is a lack of response due to the synthesis by the patient of antibody against this molecule. Because this is a molecule, this is a protein, it's an immunoglobulin, and can be attacked by our own immune system. So these ADAs, these anti-drug antibodies, can be neutralizing or non-neutralizing. Here you can see what it's a non-neutralizing. This is etanercept, or TNF. This is the molecule TNF that is blocked by etanercept. But this antibody can interfere somehow, not with the binding site, but with some component of the antibody. And here this is fully neutralizing, because you see that the active site, the binding site to the TNF, is blocked by the ADA, the anti-drug antibody that the patient synthesized, and is a cause of lack of response. So what we do in many hospitals, people subjected to this kind of anti-TNF treatments, patients having rheumatoid arthritis, spondylitis, ankylosing spondylitis, Crohn's disease, all these autoimmune disease, chronic inflammatory diseases are treated with anti-TNF antibodies. Okay? Well, so what we can find in a patient, we can find a primary failure. The patient doesn't respond at the very beginning. So then we may think that we have to change the target, right? Because the disease is not caused by this target. So it's better to use, for instance, another cytokine to be targeted. Not TNF, not tumor necrosis factor, some other drug targeting another target, another protein, another cytokine. But what is usual is that the good thing is that many people respond, and there are also patients that after some years they start not responding, and that in that situation it's. What is usually done is to measure the drug levels, the drug levels in a blood test, in the serum, to see if they are high or they are low. If the levels of the drug, the anti-TNF, are high, again, it's not acting. It's not clear, so we have to shift to another target. We have to change monoclonal antibody. But if they are low, it's important to measure these anti-drug antibodies that we have created against the anti-TNF. In that situation, we have again two possibilities. If these ADAs are high, that means that we should shift to another anti-TNF. Okay? We have to shift to another because it's not effective. It's not a matter of shifting to another target, but to shift to another anti-TNF monoclonal antibody. And if they are low, if these ADA serums are low, then we have to increase the dose. It's not a matter of the synthesis of antibodies against the drug. It's more a matter of the patient doesn't get enough anti-TNF. So we have to increase the dose, administrating more dose of anti-TNF to the patient, or shorten the interval. So this is an algorithm that is used in many hospitals. You have to measure both the anti-TNF and both the antibody against anti-TNF. Okay, this is some example of about this topic. It's one paper we published in which we wanted to see if the genes coding this Fc receptor can be responsible for the generation of ADAs. And what we saw was that this genotype that is present in the gene coding this receptor was associated with more generation of anti-ADAs, anti-drug antibodies. So before administering the drug, we can, by a genetic test, predict if this patient is possibly not going to respond because a secondary mechanism of resistance by synthesizing antibodies against the drug. As I told you, the list is not only the application of monoclonal antibodies are not restricted to chronic inflammatory or cancer. It can be used in many types of diseases, and this list is growing. I just wanted to show you something about chronic inflammatory, and this is very focused on TNF because this cytokine is crucial for the development and the destruction of the cartilage, causing the typical clinical manifestation of rheumatoid arthritis. And there's several biological treatments. Most of them are monoclonal antibodies for that diseases. And when one may think about important question about to what extent these monoclonal antibodies are similar to our natural immunoglobulins. What is the molecular basis for the inter-individual variability? Because there are patients that respond, patients that do not respond. And if we could predict the efficacy by pharmacogenetic testing, this is very important. Why? Because these treatments are very expensive. These treatments are chronic, so it costs a lot of money for the national healthcare system. So it's good to know if the patient is going to be a good candidate and will respond to the drug. And probably there is a common mechanism of the MAPS, monoclonal antibody, irrespective to the type of disease, because the Fc portion, all the effector functions of the antibody are the same. These are all immunoglobulins in most of the cases. So what is happening after the antigen recognition will be kind of the same process, and it's the immune system associated process. So this is a challenge because we are playing with the immune system. So if we want to know how these drugs, these monoclonal antibodies work, we have to understand, we have to know how the whole immune system works. So it's a challenge for for the scientific community, for biomedicine. Okay, so just trying to focus a little bit on this receptor because it binds to the FC portion of the monoclonal antibody, and there are also in some cancer, in lymphoma, it has been described that a polymorphism is associated with less response to rituximab, which is an anti-CD20 monoclonal antibody. We saw kind of the same in another disease, so there's like a common mechanism of action in chronic inflammatory diseases. We see that this, we saw that patients having this genotype respond better. They have better disease activity index in ankylosing spondylitis. We can also study the apoptotic genes by looking at polymorphism, which can help us to explain why one patient respond better than other. And here you can see some of the candidates, because as you can imagine, this way of inducing apoptosis is from the extrinsic pathway. We saw it in the neoplasia topic. So, and this is highly dependent of the activity of that receptors. So you can also study, perform some genetic testing about that. This is another study on this study genes coding that receptor or ligands, where we could see genotypes associated with good or moderate response. Another one. And in cancer, we have a lot of application for monoclonal antibodies. Why it's so important? Because it enhances the effects of conventional chemotherapy. The side effects used to be lower than for chemotherapy. They can attack the micrometastases, compensating the limited accessibility of MAPs to solid primary tumors. That's important. It's something where the monoclonal antibodies cannot penetrate the cells and cannot penetrate like a tumor, but it can bind the surface of the tumors and start inducing the destroy of tumor cells. And we can use this, and this is important, we can use these monoclonal antibodies to label, to put like a flag in this cell to be further attacked by other means, with more potent, with stronger drugs. This is a list, very simple one. Actually, it's much longer. A little bit of trastuzumab, because we have seen in neoplasia topic that here too, it's a tyrosine kinase receptor that is amplified in a subset of breast carcinoma, and it participates in a pathway inducing apoptosis migration. And here you can see that the main alteration is copy number variation. It amplifies, especially in breast cancer, and you could see it here, and it defines a subtype of breast carcinoma with a specific molecular signature. Okay, and the good thing is that it can be detected by immunohistochemistry. Here you can see a breast carcinoma with this, which is not here too. It is not from this subtype, so it doesn't overexpress HER2. But here you can see that it overexpressed. You can see in the membrane of all tumor cells the high expression of HER2. So this woman is a good candidate to be treated with a monoclonal antibody targeting HER2. That's how it is diagnosed with probes against the DNA, where we can find many copies of HER2. So this is a good candidate. This is a good candidate. Another example, and a very growing list of monoclonal antibody, is occurring in hematological mutation in lymphoma and leukemia. And the most important one is anti-CD20, sorry. CD20, the antigen, is a protein expressed by B cells that are naive and mature B cells. So we can attack tumoral B cell from lymphoma, from major B cell lymphoma. So the way this monoclonal antibody induces the deaths of tumor cells are, as I told you, these three mainly: the complement-dependent cytotoxicity, also antibody-dependent cellular cytotoxicity, and also inducing the apoptosis of the cell. This is some study we perform also by looking at polymorphism in genes coding that receptor that were again associated with response. So we could predict by a genetic test if the patient are going to respond to that therapy. Okay. So the main monoclonal antibody, anti-CD20, is rituximab, and has changed quite a lot the clinical outcome of lymphoma patients, non-Hodgkin lymphoma, especially follicular lymphoma. But there's now a second generation of maps, like ofatumumab, and that, as you can see, it's also an anti-CD20, but the target region is another part of the CD20 molecule. It's not the rituximab one, but it's another binding site. So it could be useful in case of resistance to that part, to rituximab. And in the last part of the talk, I'm going to tell you about new strategies to improve the efficacy of monoclonal antibodies. The first one is that we can improve the recognition of the monoclonal antibody by those cells expressing the receptor for the Fc. Okay, how can we do that? By a recombinant DNA-inducing point mutation that makes this affinity to the receptor higher. We can also modify the glycosylation because these immunoglobulin have a specific pattern of glycosylation. They have sugars in specific positions, and we are going to talk something about that. This is an example of inotuzumab. It's a monoclonal antibody that has glyco-engineered. It has this glycosylation in the specific site, which favors the affinity to the effector cells having the receptor for the Fc portion. Okay, so we can improve this recognition, this affinity, and make this effector cell more effective. Some other thing we can do is that, as I told you, we can label the things, the cells with an antibody, because these cells might express a wrong or altered molecule. So why don't we use toxins attached to the monoclonal antibody to kill that cell more specifically? And that has been done, and you have different approaches, different kind of toxins. Some of them are from plants or from bacteria that has to enter the cell to become active. Otherwise, it will be very harmful for the patient. So the idea is that I have a conjugate of an antibody with the drug. This is the antibody, and here you can see attached the drug. The antibody is going to recognize wrong molecule, let's say, the target, and this complex will be internalized by the target cell. In that circumstances, and under the lysosome, these components will be cleaved will be broken, and the drug will be developed, will be released into the cytosol, and this can act and kill the cell from the inside. That's the idea. So, based on trastuzumab, this anti-HER2 used in breast cancer, we can add this component, this toxin, maytansine, that is able to block microtubule formation. Is it very important for tumor cells to the microtubule? Is it important? Microtubules are part of the mitotic spindle. So every cell that wants to divide or proliferate needs to arrange the microtubule. So it's crucial for the tumor cells to have a competent microtubule. But with this drug, will attack proliferative cells. But more specifically, because it will only bind those cells expressing HER2. So we have like a double check. And it's the survival is slightly superior to just trastuzumab without the toxin. This is another one, anti-CD30, which is used in anaplastic non-Hodgkin lymphoma. Okay, and again, it has a drug bound to an antibody. And these are already used. Brentuximab is another one, also interfering with the microtubule. No problem with that. And I want, in all these strategies, I want to put like an example to make it easier to understand. So this strategy of adding toxins to the monoclonal antibody would be like the story of the horse of Troy. The horse of Troy wasn't harmful, wasn't lethal outside the city of Troy, but when it entered the city, it released from the belly all these armored warriors, that destroy the city from the inside. So that's kind of the same concept. So we have the monoclonal antibody, and inside there is a drug, and the drug will be active only when internalized into the cell. Some other thing we can attach to the monoclonal antibodies are radioactive isotopes. You know that radiation has been used in medicine for many years. In fact, it's used. The iodine, radioactive iodine, is used for the treatment of thyroid cancer. The problem is that this isotope decay very rapidly. There's toxicity. The amount of isotope that get into the tumor is very low and requires a physician. But there's also change in the emitters to make it more effective. But the idea is that you can reduce a lot the radioactive isotope by combining to a monoclonal antibody. And what is more is that you will find what is called a bystander effect. Okay, what is a bystander effect? Let's see. For instance, let's say that rituximab is an anti-CD20 monoclonal antibody against non-Hodgkin lymphoma. Okay, so this antibody will kill this cell, and this cell. But what about the rest of the cells that are not bound to the antibody? They will die. But if we combine the monoclonal antibody, radioactive isotype, then this isotype will kill the rest of the cell, and that is called bystander effect. Not all cells have to be recognized by the antibody to be killed. So that's the idea. And there's this ipilimumab tubcetan. This is like rituximab, but with a radioactive isotype. And it is another one for treating prostate cancer. Again, you can see this yttrium, this isotope in the middle. And the example in this case would be like, if you want to destroy this bridge, let's say a tumor, you don't have to put bombs in every column, in every pillar of the bridge. You just have to destroy a few of them, and then the bridge will collapse, and the tumor will die. So that's the idea. And okay, since we are playing with the immune system, we can also combine, we can attach the antibody cytokines, immune molecules that are involved in the immune regulation. Why not? And this has been done, and the idea is to increase the concentration of the cytokines in the tumor, preserving the function of both antibodies and cytokines. So here you can see, for instance, an antibody combined with interleukin 2. So when this is cleared, it will release this interleukin 2 that will attract other immune system cells where the tumor is, or the tumoral cell. Right? You can see here, and then cause the expansion of these T cells recognizing the antibody, sorry, the antigen, the tumor antigen. Do you understand that? Okay. This is a little bit more complicated because we have been talking all the time. This monoclonal antibody will recognize just one antigen, but we can synthesize what is called bispecific antibody. That means that it has two variable regions recognizing two different antigens, and we can design that. We can, the idea is to use the antibody to attract two different types of cells. One, the target cell, and then the effector cell, the immune cells that have to kill the tumor cell. You will see it with some examples. This specific antibodies, here you can see, they interact, in theory, monoclonal antibodies with Fc activate T cells in a non-specific way, and that generates toxicity. On the other hand, those mAbs lacking, that they don't have the Fc part, have lower half-life. But in bispecific antibodies, we get rid of Fc portion. They just have the variable region, but we can attract different kind of cells. If one part of the variable region is able to recognize CD3, we'll have the presence of T cells. We can attract T cells that are important for killing the target cell. We can use CD64, and a variable region anti-CD64 to attract, recruit granulocytes and even NCK cells with CD60. So here you can see this is a bispecific antibody. So you can see that it's very simple. It's a variable region recognizing, for instance, CD16 and another CD19. This is a tumor cells expressing CD16. It will be recognized by that. But the other part will attract a natural killer cell. So we are putting together a tumor cell and an immune cell that will kill the tumor cell. That's important, no? That's how we can play with this immune system. Okay, we can decide, for instance, if we want more like a T cell response, we can, instead of anti-CD16 and anti-CD3, and then we have a T cell connected with the target cell. And so this is the induction of apoptosis can also be done with these specific antibodies, and there are drugs that are used based on this concept, like this BITE technology for treatment acute leukemia. So this is quite important and emerging. Only this kind of treatment. There are some of them are approved and are of use in the clinical practice, like Binatumumab. You have to know what's the principle of this drug. It's important. It's not research. It's something that is of clinical use. Okay, this is also another specific targeting carcinoma, because we have been talking about leukemia and lymphoma. But if you put an anti-EpCAM, and EpCAM is spread by the carcinoma, you can also kill carcinoma cells. So, and the example will be like that. You know, the P-specific antibody procures the attraction of the two rivals to make the tumor cell, the immune cells win. You know, and it's like a linker between these two types of cells. Okay, and this is slightly more complicated, but it's important because there are very interesting results being obtained with this kind of drugs, what is called chimeric antigen receptor T cells. These are not just monoclonal antibodies; these are in fact they are T cells that are engineered. Okay, but the idea is to use a variable region or a monoclonal antibody linked to all the components that T cells need to be activated, right? And this only can be done by the use of recombinant DNA. So this way we can convert the humoral immunity into cellular immunity. Because we have seen in class that cellular immunity is the good one for targeting tumor cells. So antibodies can serve as anti-neoplastic drug to a certain extent, but everything will depend in the end on T cell. T cell is very important, the cellular immunity. So the idea of T-CAR cells is that we have to extract first the T cells from the patient. We have to isolate T cells from the cancer patient. And then these cells, on a plate in the lab, we treat it with a virus that contains a gene having this part coding for the variable regional monoclonal antibody and also co-stimulatory and regulatory proteins that the T cells need to be activated. I'm going to show you some other slides. I think it will become clear. So once this cell, outside the body of the patient has been treated, will be again injected into the patient, and these cells will destroy the tumor cell. Why? Because it has the variable region of a monoclonal antibody. It's a very good product, let's say. It's a very good product. Recognize the tumor antigen, and then this is a T cell. T cells expressing like a monoclonal antibody. That's really weird. We cannot find that in nature. So that is because this is engineering. It's biotechnical engineering. And here we have the proteins that are involved in the activation of T cells. So in a natural way, T cell will require the T cell receptor and some co-stimulatory molecules like this one. With this CAR technology, this engineering technology, we just take the variable part of the antibody and fuse it with this component. Here you can see one after the other, back to back. Right? And this molecule is very effective for killing cancer cell because it uses the recognition of the antibody, but the rest will be cellular immune toxicity. As I said, the process is quite complex, and it can be done in any hospital. It has to require specific facilities to first take the leukocyte from the patient, isolate the T cells, and then treat this T cell with this non-lytic virus. It's non-lytic, of course. And these cells will express, will be enriched and expand to have as many as possible, and then will be again injected into the patient along with the chemotherapy. This is another example of the same thing. And the example here is this poster. I don't know, I'm very fan of movies from the 80s, and I remember, you were born probably, I don't know, I remember the slogan of this, it was part man, part machine, all cop, all this man, no? So in the case of car, that would be like, part would be like T cell, the other part will be engineering, but all car. So it's, let's say, robo car instead of robo car. Yeah, it's would be better. Yeah, and the last strategy, it's called pre-targeting. Pre-targeting, and it's also based on this concept I told you previously, that you can put a flag on the cell. You can label the cell to be treated afterwards. So why? If we combine the monoclonal antibody with an enzyme, so imagine the monoclonal antibody and there is an enzyme, a protein with catalytic activity. Okay? This enzyme can activate a prodrug, can convert a prodrug into a drug. And this drug will only be effective in the cells expressing the antigen. Do you understand that? Yeah, we are very tired. Yeah, this guy has been talking too much on monoclonal antibodies. It has the concept. It has multi-step. So, first, imagine a cancer patient with a tumor that we have characterized. We know the molecule we want to bind, we want to attack, and so we administer a monoclonal antibody against the tumor. First, we wait a while till non-bound monoclonal antibody is washed away, is cleared from our body. It's excreted. Only monoclonal antibody attached to the ligand, to the tumor cell, will remain. And then afterwards, we administer, we inject a cytotoxin that can only be captured by the monoclonal antibody. Now you understand it better? So, what kind of toxin we can use? We can use either radioactive isotope, we can either use prodrugs. But these two components, these toxins, will only be activated because of the monoclonal antibody. So it's the ideal chemotherapy. We don't have to give a drug to the whole body of the patient. This is crazy. That's what we do, but this is crazy. If you want to treat a tumor, go for the tumor, not go for the whole body. So what we administer is not a cytotoxin. It's a prodrug. It's not effective unless something activates it. And the only thing that can activate this drug is the enzyme that is attached to the monoclonal antibody. These are some examples of that. The idea is to use something. The concept is very based on what we have been using for many years to detect proteins in immunoassays. Either ELISA, Western blot, immunofluorescence, immunohistochemistry, they are all based in that concept. There is an antigen we want to test in a sample, and we use a primary antibody targeting this antigen, and then a secondary antibody with an enzyme. We put the substrate, and the enzyme will make the substrate glow, fluorescence, or become visible. Okay? And that's all. So the conclusion of this seminar, hope you it will be clear for all of you, is that this monoclonal antibody constitutes a fast-growing and emerging strategy in biological therapy. Their mechanism of action is not fully discovered, it's not fully known, because it's as complex as the immune system. And it can be applied to many different diseases, and it's changing our idea of therapy. I hope these messages are clear for you, and that's the end of the seminar. If you have some questions? Yeah, you can.
Lo que has dicho que el isótopo relativo que solo se activa cuando se une al pulmón, también se podría utilizar como diagnóstico. Si tú le unes a un radioisótopo que se pueda ver por un TAC, por ejemplo, claro. Si solo se une siempre a todas las células tumorales, también podría ser para... Claro,
lo importante es que tú, precisamente eso, que seas capaz de eliminar, de que el anticuerpo se elimine del cuerpo para que solo el que esté unido, porque si no verías todo el cuerpo.
No, claro, que como se elimina, eso aparte de ese tratamiento también podría ser para delimitar bien las cavidades del tumor y poder también operarlo.
Claro. De hecho, o sea, la parte diagnóstica ha ido siempre por delante. La parte terapéutica no ha ido tan rápido por el tema que hemos comentado al principio de la inmunogenicidad, de los problemas de efectos adversos también que tienen los fármacos por el hecho de ser biológicos, pero claro, se podría hacer. Lo importante es que se busque alguna manera de que ese isótopo esté de alguna manera, le llamamos puenteado o pantallado, para que solo emita esa radioactividad cuando está unido al anticuerpo. El anticuerpo está unido al antígeno. ¿Alguna pregunta? Pero yo creo que los de la siempre preguntan mal, no sé.
Estamos
pero yo creo que en general, bueno, sé que era larga, las cosas complicadas, pero yo lo que quería también era que les perdierais el miedo a una clase de inglés. O sea, más o menos se sigue, ¿no?
Sí.
Esa era un poco la idea, pero eso también, lo que he dicho también. Esa era un poco la idea, ¿vale?
Sí. Gracias.